So now we continue with the program. It's my great pleasure to introduce our next speaker, who is very well known to everybody here because she's been at UCSF and is also world famous, at least nationally, certainly nationally famous, advisor to presidents and all that sort of stuff. Cori Bargmann.
Hi everyone. This is such an emotional occasion. It's a sad occasion, but it's also an occasion for celebration, for talking about what really matters to us and reflecting on what matters to us. And I came to UCSF a year or two before Allison did, and I came with the following idea or plan, which was that the human brain was much too complicated to study, but that ultimately it was made of genes and not very many of them. And maybe by studying simpler animals that shared many genes with humans, we would be able to understand important things about how the brain worked, and in particular, how different genes contribute to the functions of the brain. And what I hadn't really anticipated is the extent to which this other brain here, the songbird brain, would really be teaching us everything from the larger to the smaller levels about how brains work. And I've actually thought a lot about Allison's work and her impact. Her impact was so tremendous. And beyond that, even the songbird field, considering the number of people who are in this field, has had a really disproportionately large impact on our understanding of how brains work. And thinking about what sort of intellectual aspects and conceptual aspects have really led to that sort of increase in Allison's work and the way that it's affected the field as a whole, using my own work as a small example of that. So we've all been sort of showing the point at which we imprinted on Allison as our first real experience with her. That's appropriate because, of course, birdsong is an early life experience that then leads to lifelong changes. And this slide here shows the first time I really spent a substantial amount of time with Allison. It was right at the time that she started at UCSF, so over 20 years ago. And we are actually in Japan. We're at a workshop on neuroethology in the brain. And the reason I want to show this paper is that I feel like people have been talking about Allison as a scientist and Allison as a person, and for me those two things are completely entangled in one another. I just don't see how you can separate them from each other. And here we were at this meeting in Japan, and it was a scientific meeting, and we were discussing neuroethology and behavior. But it was also a human experience. We went around Tokyo. This is in Asakusa. We went to a karaoke bar in the Ginza, which was one of the most degraded human experiences I have ever been exposed to. We did sing. We went to Kyoto, and the other thing I love about this picture is that this is also the very first time that I met Michael Brainerd, who was still a graduate student at Stanford at the time. And so you can see that, in this one meeting, all aspects of her life were really coming together already. Her scientific life, her enjoyment, her openness to new experiences in new places, and the attachment to her family and to her loved ones. And so returning to the lab, here's the problem that my lab set out to study, is to really try to understand how the sense of smell works and how the sense of smell is linked to behavior. And the sense of smell is an ancient sense that animals share, and so the tiny worm has a sense of smell like people do. And we did genetics on that to try to understand its underlying basis, and we found, for example, that there were many, many, many G-protein coupled receptors that sense odors that allow the worm to sense the world. And we could find specific receptors, like this guy here, for the specific odor diacetyl, and that let the animal do a behavior. It let the expression of this receptor allowed the animal to sense diacetyl, to buttery odor, and to move toward a source of diacetyl from a starting point. And these are a bunch of little worms making up their minds. But then we did an experiment. This was done by Emily Tremmel, a graduate student in the lab, where she used the genes to ask a different kind of question about how the brain worked, where she took this G protein-coupled odorant receptor and she moved it from its normal location onto a different neuron. And this allowed us to selectively interact with a different neuron, but with the same odor. And what she found when she did that was that the animal responded to the odor, but it now generated the exact opposite behavior, that there was an avoidance of this odor instead of an attraction. So we had somehow moved from thinking about the biochemistry of detection of odors to something about how the brain encodes innate odor preferences. And we realized right away that that was really a much more interesting and important question. And so we tried and have spent a long time trying to understand how that works. And in fact, C. elegans was the ideal place to do this. It already had what was called a connectome, a map of all the connections of the neurons, of the 302 cells in its brain. And we could start from our neuron of interest, which is in here, and try to understand everything else that it was connected with. But we actually found it very difficult. And we found it difficult to make progress on this problem of linking neuron to behavior beyond the sensory neuron for a very long time. And I think actually one of the most important conversations I ever had was a conversation with Allison. It was not actually about this problem, but about a different problem we were working on in the lab. And she said, why is it that when you talk about genes and molecules, you're so precise, and when you talk about behavior, you're so vague and impressionistic? And it was a gigantic revelation to me. And the funny thing about this is I reminded her of this conversation a year or two ago, and she'd completely forgotten it. But that's because it didn't change the way that she thought about things. Because this is one of the great things about Allison's own work. And this is one of the reasons that birdsong has been such an important system in teaching us about the link between sensory inputs and neural circuits and behavior, which is that you think about these problems in a very precise way. And this is a paper from Michel Solis's work that just kind of illustrates a set of different aspects of that sort of quantitative and precise thinking that allowed Allison to make such great progress. So first of all, you have to be able to manipulate this. This is a particular experiment about how the neuron responds to the bird's own song versus the song that it learned about that Mimi told you about, the tutor song. And in order to distinguish those, actually, you had to do sort of a clever experimental manipulation to make those different, because normally they're very similar. So you had to sort of change the input into the system. And then you had to play with the different sensory signals. You had to do things like play signals in reverse. And then you had to quantify what came out of that, these spike trains, into just not yes or no, but really into kind of really analyzing what kinds of distributions occur. And these are things that you've heard from all the talks today. But these were not things that were commonly used, and they're still just invading the sort of more genetic organisms like worms and flies and mice and even humans. I think this sort of intuition of really controlling the stimulus and measuring the behavior and measuring the intermediate stages is so important for making progress. So in terms of the problem that we were trying to study of chemotaxis, the way that we started to make progress on that was to try to do exactly this, was to try to understand the behavior and to manipulate the sensory input so that we could generate the behaviors under controlled conditions. And we started with an observation from Sean Lockery's lab that said that one of the ways that the worm approaches an odor is by regulating the probability of a turn. That basically when it's moving in the right direction, it just keeps moving, and when it's moving in the wrong direction, it changes direction, which is not a bad life strategy either. And so as a result, you end up with long runs in the right direction and short runs in the wrong direction, and you end up where you want to go. So what we set out to do was to try to develop a way of really generating the stimuli that regulate this behavior and understanding how the animal responded to those stimuli and how the worm's brain responded to those stimuli. And this involved a lot of work by an engineer in the lab, Dirk Albrecht, designing very precisely controlled microfluidic environments that let us expose a freely moving animal to different kinds of odor patterns that we would specify. We could give it a temporal pulse, so just odor for a few seconds and then take it away to give it the illusion that it had gone into the odor and then came out. We could give stripes, we could give gradients, and then we could monitor and videotape and quantify the behavior of these animals in these environments. And moreover, as tools became more advanced, we have been able to actually monitor the activity of individual neurons in these animals while they're doing this task. And so this one little bright spot in the worm's head is a single olfactory neuron expressing a genetically encoded calcium indicator. And as the animal moves around in this little arena, we can track when odor is present, when odor is removed, what the animal does here, and what the neuron did. And so we're looking here that actually at this point here, when odor was removed, the animal did a little turn or reorientation, and that was associated with an increase in calcium levels in this olfactory neuron. So what do you get out of this? So our prediction is, as you saw here, that when odor is removed, the animal should turn. And what we see is that on average, that's true. Okay, so on average, when you remove the odor, about 2/3 or 3/4 of the animals make a turn. But in fact, there's a lot of variability in this behavior. A single animal's behavior varies from trial to trial. And so here, actually, we looked at 23 animals, and we gave them 24 stimuli, and every single animal responded sometimes and not others. And you can work out, one thing you could do is to just sort of declare victory with 70% of the animals responding and feel really good about that. But actually, here's another thing that I learned from Allison, and this is something that Steve Lisberger alluded to earlier, is that Allison was one of the first people to really think carefully about the fact that variability is a real property of behavior. It's not just a passive property. It's an active, creative property of the way that behavior is generated and even that learning occurs. And so you shouldn't just ignore these things and average together and say, well, the animal reversed 70% of the time. You should try to figure out what's going on there. And this is illustrated here. Several people have referred to Neil Hessler's work with Allison, showing the importance of variability of neural responses and the fact that it's actually actively regulated, not just a limit of the system. So what was nice about the worms is that there's so few neurons that you can really focus that problem down to a small number of components where, if you don't have an explanation, you don't have anywhere to hide, okay? You can't just blame some other part of the brain. So here's our olfactory neuron that can regulate turning. And down here is a neuron in the worm that's called the backward command neuron. And that neuron drives reversal behaviors. And the fastest way to get from point A to point B is actually through this neuron here. But it turns out that there are also very strong neurons between these two neurons here. So this level here has been classically considered the level of sensory integrating neurons. And then these are neurons involved in regulating different kinds of motor behaviors. And so where between the olfactory neuron and the olfactory integrating neurons and the backward command neurons does this take place? And I'm just going to emphasize here these 4 neurons, but just remind you that there are many more neurons in this circuit. So the first question is based on that little movie I showed you. We can look at a single animal responding to odor many, many times. This heat map here shows a single animal responding to odor 80 times, once every 20 minutes, over a 2-hour period. And each of these lines represents what this sensory neuron did every single time the odor appeared. And every single time that the odor appeared, its activity went down a little, and every single time that it disappeared, its activity went up a little. So basically, these hot colors here indicate that the neuron really seemed to respond. The worm knew that that odor was there, or it wouldn't have generated this signal. So, but the really interesting experiment is not even that so much, but relating it back to what the worm did every single time that the odor was there. So we can separate out those 80 trials, and on about 20 of them, the worm did a big reorientation behavior, and on about 30 of them, he did a simpler reorientation behavior, and about 20 of them, he just ignored it completely. And so then you ask, okay, now let's separate out all these trials and ask what the sensory neuron did every single time. And the answer is every single time the sensory neuron did exactly the same thing. The olfactory neuron is in no way predicting what the animal does. If you take these 3 curves and put them on top of each other, they are identical. So this is not where the behavior arises. So we have to look further down. Okay, let's look all the way at the bottom here. If we look at this backward command neuron here, and this is a result that's come from many labs, not just ours, we find that its activity is basically perfectly correlated to when the animal reverses. 90+% of the time the animal reverses, this neuron is on. So here we're at the behavioral output. We can look at this neuron and basically tell if the animal's reversing or not, and that's different from the sensory input. So now we can simplify the problem here to looking at the steps in between to ask where the variability arises. Where does variability first show up? One place where variability can happen is at sensory output synapses. This is a very active site of regulation. For example, David Anderson's lab has worked on this, showing how this can be a site of regulation. And so we looked at that using a set of different molecular tools that let us look just at the axon of the sensory neuron and just in detail, either at the calcium levels within the axon terminals or at the release of synaptic vesicles from the individual sensory neuron. And basically from both of these, it seemed like these synapses are reliable under these circumstances. They're not changing from trial to trial. So this neuron not only recognizes what's happened, but apparently reports what's happened to the downstream neurons. But when we move one step down, when we move to this very next sensory integrating neuron here, one synapse away, we find that the reliability goes down. So instead of showing this sharp, very strong suppression and then enhancement in its signal, it shows a lesser enhancement. And I have to say, this is when you take all the responses and average them. And this is what my people do. We say because, central limit theorem and stuff. But in fact, if you listen to Allison and you think about what you're really trying to solve, you have to go back and look not at the average response, but at the individual responses. And by looking at the individual responses, you can right away see that there's something a little bit strange about these heat maps, which is there's a lot of red and a lot of blue, but no yellow. And what that means is that even though this here is kind of an intermediate response, the individual responses are not intermediate. They're either yeses or nos at the level of these neurons here. And you can actually quantify that. You can divide out the different tracks in the heat map and ask what happened. And what you find is that the sensory neuron responds 100% of the time. And this first interneuron here responds 62% of the time, almost as well as the sensory neuron. Bang, red to blue. We're just going to look at this first part. But then the other 40% of the time, it's kind of all over the place. And then when you get to the backward command neuron, you're seeing it responding about half the time. And this is about what you would be seeing in terms of the reversal behavior. So you're seeing kind of a loss of reliability along this pathway. So this gives us a sense of where this is happening. It's happening between here and here. So what else can we learn about this? Again, looking at the individual responses tells us something. For example, looking at the individual responses of this neuron here shows us that this neuron has bistable states. It's either got high activity or low activity, but it seems to jump between them very rapidly, both in the presence and in the absence of odor. But what we really learned something from was looking not just at this neuron, but at all of these neurons simultaneously. And this is done by labeling each cell individually with a genetically encoded calcium indicator and watching them in response to odor or on their own. And what you can see immediately is, first of all, they're all bistable. They all jump between these high and these low states. And second, most of the time they jump at the same time. These three neurons are correlated with one another in terms of their activity. So we can go through many, many hours of movies like this, looking at the activity of these neurons and analyzing them in different ways. And basically what you find is that if you have three neurons, and they can be either on or off, that gives rise to 8 different possible states. But only 3 of those states are ever observed. There's a state in which all 3 are on, a state in which all 3 are off, and a state in which only the downstream interneuron, right downstream of the olfactory neuron, is on, and the others are off. So armed with this knowledge, we can go back to and ask how these neurons might be responding under these different circumstances. We've identified different classes of events that we can now look at separately. So let's start by looking at the point where all 3 of them are on together. When all 3 of these neurons are on together, the odor responses occur more or less half the time, and they don't occur the other half of the time. So they're unreliable. In general, if one of these neurons responds, all three of them responds, in this individual case, as in the earlier ones I showed you. But then we can pick out the subset of the time when only this guy here is on and the other two are off. And when we look at that subset of the time and we look at the heat map, the neuron responds to the odor every single time. So there's reliability in these responses under some circumstances. There's some kind of a pattern of ongoing activity that we can use to predict whether the neuron is going to respond or not. And that activity correlates with what's going on in downstream neurons in the circuit. So that's a correlation. One of the nice things about the system we work in is that it's possible to actually directly manipulate neurons of your choice in various kinds of ways. The ones that we chose were to use a neurotransmitter receptor that does not exist in the worm but exists in the fly. It detects histamine, and it silences the neurons in which it's expressed. And when we express this receptor in different worm neurons, that makes those neurons responsive to histamine, so we can give sort of a false transmitter and change their activity. And that lets us directly manipulate the circuit. So for example, we can generate exactly this activity state that we observed was correlated with responsiveness. We can take this neuron, even though a lot of the time it's normally active with all three together, and we can shut these two guys off, and we can add odor and ask, now what happens? And the answer is, it now responds 90-some% of the time. So it's now become much more reliable. So this activity state is not just correlated with the response to odor. This activity state appears to be sufficient to make a neuron reliable. And furthermore, we can also, with this kind of experiment, generate activity states that never exist in nature, right? We can generate false states. And one of the states we can generate is this false state here, in which we only silence this side neuron here. And the side neuron here was kind of the little bit outside of the normal pathway, but what was really striking is that when we took this guy out, all of a sudden, this neuron also became very, very stereotyped, very reliable, as reliable as if we had shut the whole thing off. And moreover, all of a sudden, this guy here has also become much more reliable. So we think of this neuron here as kind of Loki the trickster, who's generating conflicting information in the circuit to keep these neurons from responding to odor, which of course is what they should be doing, not the neuroethological point of view here. But generating a different kind of signal. So these are all activity patterns in a circuit. But what's nice about this is that we can then return to the behavior, and we can look at the behavioral responses to odor. And so this experiment here just shows you an experiment where you look at the animals and their sort of reversal versus forward behavior before odor is added, when odor is added when they move forward, and then when odor is taken away. And if you silence the RAM neurons, not only were the circuits more reliable, but the odor response is more reliable. Compared to a wild type, which sort of meanders up in this axis and meanders down, these guys shoot up and then they shoot down again. Okay? So this neuron is actually generating variability not just in the neural responses, but in the behavioral response to an odor. So the conclusion of this is that the way that variability is generated, if we think about this from the point of view of this integrating neuron here, what seems to be happening is that odor and a motor circuit are actually competing to control this intermediate neuron. So this neuron here can respond to odor, which it does very reliably when it's on its own, or it can respond to the overall state of the network, what the other neurons are doing. And because of that, when this and the site of coupling of this neuron to the network is this neuron here in the intermediate, it seems to couple these three neurons together to couple them to a reversing behavior state. So here we can move from an olfactory neuron to a backward command neuron to a set of more variable responses where variability can give rise to different behaviors at different times. But a different way of kind of looking at this result is actually to sort of, again, return to the songbird and really what I think was one of the really profound conceptual insights of the songbird system and of Allison's work in particular. And that is that, one way of talking about what we found is that sensory circuits, the very first neuron even downstream of a sensory neuron, is already part of a motor circuit. And really, one of the major lessons from the songbird system is that these neurons, which respond in a sleeping bird to just hearing the bird's own song, in an awake bird are activated when the bird is singing by the motor pathways that drive song, that a sensory circuit is a motor circuit, and that our division of the brain into these two different areas is really artificial and not the way that signals really work. And when I look at some of the things that are happening, I think this idea is really moving further and further. This idea of sort of coherent and linked functions is moving into more and more systems. Michael Stryker's recent work in the visual system showing inputs from motor systems, for example, really gives you the same idea, and that understanding the relationships between these things is really important. And just to mention at the end that I've shown you a subcircuit and a much more complex circuit, of which there are many other pieces that surely are left to understand. So I'm going to switch now one of the points of this actually maybe is to bring up another point of Allison's work, which is to show that there is actually adult learning as well as early imprinting. You can continue to learn from people, just as birds can continue to learn to adjust their song over time. So about a year ago, I was telling my sister, who's a reader and a writer, about Allison, and she sent me the following that Rilke wrote on birds. And he said, "We take a bird song into our own inner selves so easily. It seems to us that we translate it fully, with no remainder, into our feelings. A bird song can even, for a moment, make the whole world a sky within us, because we feel that the bird does not distinguish between its heart and the world's." Now, I'm not sure I understand this, but I really liked it, and I sent it to Allison, and she really liked it too. And here's what I think. I think Allison didn't really distinguish between her heart and the world's. I don't think she distinguished between her work and her human interests and her family. And also, I think that she was open to the world in a really fantastic way. She wanted everything. She wanted the world. She wanted to be a great scientist. She wanted to be, at the same time, a practicing psychiatrist. She wanted to be the best mentor to her students and her postdocs. She wanted to be a wife. She wanted to be a loving mother. She wanted to be a friend, and she was a truly fantastic friend. And, many of the things that Allison did, deciding to have kids at age 50, maybe that was a crazy thing to do, but it was a fantastic thing to do, and it was taking the world into your life. And her devotion to them and her continued devotion at all levels, here from a picture from last year, visiting Allison as late as last September, she was talking about, problems with the searches that she was chairing at UCSF and what the latest cool results were from Hamish, a postdoc in her lab, as well as everything else. And you might think, well, people might choose to put some of those things aside, but she didn't. She wanted everything. She wanted it all together. And her friendship was really fantastically valuable. This is Gail Martin here. This is one of our Don Ganem-sponsored Girls' Day Outs. There were other Girls' Day Outs. People have referred to the Saying No Club, and Luann Brizendine actually used to put on these girls' night outs where people would go together. She was there for the good times. She was there for the bad times. And, I want to pick up on something that came up early this morning about Allison being a tireless supporter of women in science. If you want to support women in science, you should spend time with them. You should be there. You should really communicate. You should be with these people. And she was there for people. Allison knew that science was done by people. And people were the people in her lab, and she accepted them as people as well as seeing them as scientists. And she put that together. And she put that together here too. So there are people who go through life in a way that's really perfect. They set every step perfectly in front of each other. They do it with total grace and beauty. Allison was not that person. She was really messy. She was disorganized. She was always late. Her emails were a lowercase stream of consciousness that would trail off in the middle of a sentence. Her conversations sometimes had the same quality. But you know what? In some ways, when I've been thinking about this, that's one of the things I love about her the most, because she was human. She showed that you could do it and you could be human. You weren't some godlike creature from another planet. So I was trying to think about a way to express this, and I pulled up a sonnet from Robert Herrick, and I consider myself entitled to bring up sweet disorder in the dress because Allison and I shopped together, but also because she, it's actually kind of true that her shoes would be untied and her cuffs would be neglected, and the point of the sonnet is that disorder is more beautiful than order, and that all the individual disordered features of his beloved one's hygiene do more bewitch me than when art is too precise in every part. And I think Allison was bewitching, and we are all bewitched. And I just want to close on quoting another very beautiful blonde on this topic, Marilyn Monroe, who said, "Imperfection is beauty, madness is genius, and it's better to be absolutely ridiculous than absolutely boring." And I think Allison would have signed on to that in a minute. Thank you.